Good afternoon, everyone. Can you guys hear me okay? Great. I'll just start my start now. And can I just get you guys to confirm as well that you guys can see my screen? Great, sounds good. So I think we should get started. You only have seven people in the call today, but as they come through, I'll try to admit them. But just for the interest of time, given that we are going through a lot of bulk this session, I'll just start on time. All right, so the topic for today is the central visual system and also the auditory and vestibular system. So as I noted earlier, it is a pretty bulky lecture today with two parts. We'll start with the central visual system and then move into the auditory visual system after that. So as always, if you guys have any questions at all, feel free to drop them in the chat. Otherwise, feel free to send me an email, then I can answer offline as well. So just to give you guys, just to go through the different objectives that we have for the first part of this session. We'll describe the relationship between vision and perception, describe and explain the visual pathways, including the retinal input, the role of the lateral geniculate nucleus, abbreviated to LGN, the layers of the lateral geniculate nucleus, organization and the role of the striate cortex, visual selectivity, the parallel pathways, and the ventral and dorsal streams as well. So just to give you guys a brief introduction about the first half of the topic. Neurons in the visual systems are responsible for the perception of what we see in our surroundings. The pathway of the neural processing starts from the retina, which is propagated to the brain via the lateral geniculate nucleus, the primary visual cortex, and the higher order visual areas of the occipital, temporal, and parietal lobes. Neurons in the visual systems have overlapping receptive fields, which is sensitive to different facets of the visual inputs. The retinofugal projection is the neural pathway that leaves the eye, beginning with the optic nerve. To easily remember this term, fugal means to flee and is commonly used in neuroanatomy to describe a pathway that is directed away from the structure. In this case, it is moving away from the retina. We begin looking at how the retinofugal projection courses from the eye to the brainstem on each side, and how the task of analyzing the visual world is initially divided among and organized with certain structures of the brainstem. From there, we will focus on the major arm of the retinofugal projection that mediates conscious visual perception. The axons leaving the retina goes through three structures before they form synapses to the brainstem. These are the optic nerve, the optic chiasm, and the optic tract. The optic nerve from the optic chiasm is named from the X shape from the Greek letter chi. In this area, there is a decussation of axonal fibers from one side to the other side. It is important to note that there is only a partial decussation here, mainly attributed to the nasal retinas. From the chiasm, fibers form the optic tract, which runs along the medial surfaces of the diencephalon. Now, the full visual field is the entire region of the space that can be seen with both eyes looking straight ahead. If you fix your gaze ahead and imagine one point in your visual field that you can fixate on, now pass through a line dividing the visual field into the right and the left halves. The object appearing on the left side of the midline are in the left hemifield, while the objects appearing on the right of the midline are in the right side of the visual hemifield. The binocular visual field refers to the area of visual field that is detected by both eyes, and this is exemplified by alternating closing one eye and then the other. It is important to note that the nasal portion of the left retina projects axons to the right side of the brain. Similarly, the nasal visual axons that project from the right side go to the left side of the brain. This is made possible by the optic chiasm. Essentially, the left hemifield is viewed by the right hemisphere, while the right visual hemifield is viewed by the left hemisphere. Most of the projections end up in the lateral geniculate nucleus, or LGN, of the thalamus. The neurons in the LGN gives rise to axons that project to the primary visual cortex. This is also called the optic radiation. It has been shown that when a lesion is applied to the retinofugal projection from eye to the LGN, this can cause blindness in humans. Therefore, we know that it is the pathway that mediates conscious visual perception. Depending on where the fibers are cut, the retinofugal projection, this can lead to different outcomes in the perception of the visual field. As you can see in diagram A, where the optic nerve is cut, the vision of the left eye will be completely lost. In diagram B, when the optic tract is cut on the left side, the vision will be completely lost on the right visual field of each eye. Lastly, if you look at diagram C, if the optic chiasm is cut, only the crossing fibers will be damaged, leading to the loss of the peripheral vision. Axons emerging from the retina also project to other areas aside from the lateral geniculate nucleus. Firstly, this travels to the hypothalamus, which can impact synchronizing a variety of biological rhythms, including sleep and wakefulness with the daily dark light cycle. There's also a direct projection to the midbrain, more specifically to an area called the pretectum. This area is responsible for controlling the size of the pupil and certain types of eye movement. Another area in the midbrain that is innervated by the retinal cells is the superior colliculus. The superior colliculus plays a role in orienting eyes in response to a stimuli, and this is called the orienting response. So an example of the orienting response is when you when you hear an ambulance coming, you tend to fixate your gaze to the incoming ambulance. And this response is referred to as the orienting response. The primary visual area is also referred to as Brodmann's area 17, and this is located in the occipital lobe. Much of this area lies on the medial surface of the hemisphere surrounding the calcarine fissure. Other names of this area is V1 and also the striate cortex. The projection starting from the retina and extending to the lateral geniculate nucleus and V1 illustrates a general organization feature of the central visual system, which is called the retinotopy. Retinotopy is defined, it by definition refers to the organization whereby neighboring cells in the retina feed information to neighboring places in the target structure. In this case, the LGN and the striate cortex. In this way, a two-dimensional surface of the retina is mapped into the two-dimensional surface of the subsequent structures. The neighboring locations of the retina project to neighboring locations in the LGN. This retinotopic representation is preserved in the LGN projection to V1. The lower portions of V1 represents information about the top half of the visual space, and the upper portion of V1 represents the bottom half of the visual space. Also notice the distortion of the map in the diagram. It is important to note that similar maps are also found in the superior colliculus. From this slide, you can see that the striate cortex is made up or divided into different layers from layer 4 to layer 4C. These are mainly composed of monocular neurons. However, when you get to the superficial layers, mostly neurons are binocular, which means that they receive input from both sides of the eye. The receptive fields of neurons in layer 4C are similar to those in the lateral geniculate nucleus. In this case, these are monocular receptive fields. Layer 4C alpha in particular are insensitive to wavelength of the light, whereas in the layer 4C beta, the neurons exhibit center surround color opponency. Beyond layer 4C, most neurons in these layers are binocular, which means that they receive input from both eyes. This also means that neurons have two receptive fields. One is ipsilateral, while the other one is contralateral. The orientation selectivity refers to how our brain can be selective in its response to a particular visual stimulus. As you can see in the diagram, the orientation can either elicit a strong response if the visual field stimulus is of a correct orientation. Other stimuli with unmatching orientation leads to a weaker response, in some cases, no response at all. With regards to the direction selectivity, many V1 receptive fields exhibit direction selectivity. They respond when a bar of light at the optimal orientation moves perpendicular to the orientation in one direction, but not the opposite direction. Direction selective cells in V1 are a subset of the cells that are orientation selective. These neurons are thought to be specialized for the analysis of object motion. Cells with simple receptive fields are binocular. orientation selective, and have a characteristic on and off areas in its receptive fields. If you refer to diagram A, both sides are referred to as off areas, wherein the areas respond to lights turning off, while the on area mainly respond to light when it is turned on. Like cells with simple receptive fields, complex cells are also binocular and orientation selective. However, it does not have a distinct on and off regions. As you can see in the diagram, correct oriented light leads to the generation of an action potential. However, incorrect orientation does not elicit a response. Furthermore, regardless of the position of the bar of light, this leads to an action potential and accompanied by a response to off for each of the stimuli. The anatomy and physiology of the central visual pathway from the retina to the striate cortex are consistent with the idea that there are several channels that process visual input. And this can be processed in parallel. Each one of these appears to be specialized for the analysis of different facets of the visual scene. With that said, the magnocellular, the parvo interblob, and the blob pathways were distinguished. The pathways are described in this diagram. So going through each one of them, the magnocellular pathway begins with the M-type ganglion cells of the retina. These cells project axons to the magnocellular layers of the LGN. These layers project to layer 4C alpha of the striate cortex, which is then relayed to layer 4B. These cells in layer 4B have binocular receptive fields of simple and complex type. Looking at its property, these are orientation selective and many are direction selective as well. They are not generally wavelength sensitive. It is thought to be involved in object motion and guidance of motor actions. The next pathway is the parvocellular interblob pathway. This starts with the P-type ganglion cells. These cells project axons to the parvocellular layers of the LGN. These layers project to layer 4C beta and to the layer 2 and layer 3 interblob regions as well. These neurons are not generally direction selective or wavelength selective. The binocular receptive fields are orientation selective with simple or complex receptive fields. Neurons on this pathway have the smallest orientation selective receptive fields, which suggests that they are involved in the analysis of fine objects. The final pathway that we have on this slide is the middle one, which is the blob pathway. The neurons on this pathway is more mixed compared to parvocellular, interblob, and magnocellular pathways. These arises from the subset of ganglion cells that are neither M-type or P-type cells. These cells project to the coniocellular layers of the LGN, which then projects to the cytochrome oxidase blobs in layers 2 and 3. As you can see in this diagram as well, the blobs are a site of convergence of parvocellular, magnocellular, and coniocellular input. Properties of blob cells include having receptive fields that are center surround and color opponent. They would have monocular and lack orientation selectivity. And lastly, they would have high incidence of wavelength sensitivity, which suggests that the neurons are involved in the analysis of object color. The cortical module refers to a chunk in the primary visual cortex, about 2 by 2 millimeters, that is capable of analyzing or recognizing a point in your visual field. This chunk was shown to be necessary mainly because the removal of this led to an onset of a blind spot in your visual field. It was also noted that this chunk is sufficient in terms of analyzing and recognizing a point in your visual field because it contains all the neural machinery to analyze a point in your visual field. The striate cortex is made up of thousands of these cortical modules. Beyond the striate cortex, there are 2 large cortical streams that were identified. So you have the dorsal stream and you also have the ventral stream. The dorsal stream is noted to analyze visual motion and visual control of action, while the ventral stream, on the other hand, is noted to be involved in the perception of the visual world and recognition of objects. These processing streams were primarily studied in monkeys, where brain measurements from a single neuron was made. This slide shows you the different visual areas that are responsible for your visual perception. These are abbreviated as V1, V3, and V4. There are other areas not noted on this diagram, including V2, MST, and IT areas. And in figure B, you can see the location of the V5. This is also referred to as area MT because of its location in the middle temporal lobe. This area is shown to be involved in the processing of object motion. So moving on to the next half of the lecture, this section we will go through the auditory and the visual systems. So the following are our learning objectives for the second half. We will go through the structural components of the auditory system and their functions. We will describe each step of the auditory pathway from the ear to the brain. We will explain how various sound characteristics are calculated in the brain. We will look into the structural components of the vestibular systems and their functions. And lastly, we will look into the central vestibular pathway and each of its steps. And just to give everyone a basic introduction on auditory and vestibular system, the auditory system mainly responds to auditory stimuli. Audition is termed as our sense of hearing, which is responsible for detecting sound in our environment. So this could be someone in our periphery talking, playing music, or operating a loud machinery. What is amazing about our sense of hearing is that it has the ability to perceive and interpret nuances. In this case, when you are in a noisy environment, you actually have the ability to focus and discriminate between the sounds that you hear. The other side of this lecture highlights the vestibular system, which is responsible for controlling our sense of balance. This processes our location and movement in a given space. The stimuli that activates our auditory system is sound. Sound is generated by the changes in air pressure. In this case, when you have the compression and rarefication of air, a sound can be generated. The cycle can be defined by the distance between successive compressed patches of air. Furthermore, the frequency is calculated by the number of cycles per second, which is expressed in hertz. Not all sounds are considered audible, which means our sense of hearing doesn't detect all sound waves. Audible sound is mainly those that are within the frequency range of 20 Hertz to 2000 Hertz. The pitch of the sound can be defined by the frequency of the wave. In this case, when you have a high frequency wave, you will perceive a high pitched sound. Meanwhile, when you have a low frequency sound wave, you will hear a low pitched sound. The intensity of the sound is basically driven by the amplitude of the wave. And if you look at the diagram, it is defined by the degree or the size of the deflection that you see in the sound wave. The auditory system is confined in the ear, much like all the sensory systems that we've discussed in neuroscience. The activation of a sensory organ is brought upon by the activation of its receptor, which in this case is the same for our auditory system. The ear is mainly divided into three parts. So you've got the outer ear, which is composed of the pinna and the auditory canal. The middle ear, which are composed of the tympanic membrane, ossicles, and the surrounding muscles. And lastly, the inner ear, which is composed of the cochlea and its components. The outer ear serves a purpose for collecting sound waves generated in our environment. It is designed to optimize its function and allows us to detect sound and also perceive nuances of audition. The middle ear is responsible for transducing air pressure into mechanical pressure, which will allow the propagation of signals to the cochlea, which is responsible for transducing the mechanical pressure into an action potential. As we go deeply into the lecture, we will highlight how this can occur. From the last slide, you can understand the basic auditory pathway that leads to the generation of an action potential. In this case, when you have a generation of a sound wave, this gets picked up by the ear, which travels into the tympanic membrane. Movements in the tympanic membrane allows for the ossicle to move, which puts pressure on the oval window in the cochlea. And from this action, the cochlear fluid will move in response and will ultimately lead to the generation of a sensory response. The purpose of the middle ear is to amplify the sound wave that we detect in our environment. Having sound wave travel to the cochlea directly does not allow for effective transfer of signals from air pressure to the movement of the fluids in the cochlea. Therefore, a mechanism in between is needed to allow for this transfer of energy from air pressure to the movement of fluids in the cochlea. The middle ear is composed of ossicles and surrounding muscles that allow for the movement of these ossicles. Movement of the ossicles eventually lead to the stapes hitting the oval window in the cochlea, which provides enough pressure for the movement of fluids in the cochlea. There is also the attenuation reflex, that is a protective mechanism within the middle ear. This allows us to protect ourselves from loud noises by the contraction of the surrounding muscles in the ear. Though not all mechanisms are infallible, and there's a lag time between detecting a loud noise and the activation of this attenuation reflex. Therefore, there is still a risk in getting hearing loss in the onset of loud noises. Now, the inner ear is composed of the cochlea and the inner structures contained in it. If you dissect a cochlea, you can mainly see three cavities filled with fluids. The cavities are called the scala vestibuli, scala media, and also scala tympani. The scala vestibuli and tympani contains a fluid called the perilymph, while the scala media contains the fluid called the endolymph. It was identified that there is a endocochlear potential between the two fluids, where the endolymph was shown to be more positive than the perilymph by about 80 millivolts. As discussed in the previous slide, when the oval window hits the stapes, this allows for cochlear fluids to move at the end of the cochlea. There is a round window that ends up bulging once the cochlear fluids move around the cochlea, and the signal transduction within the cochlea is marked by the movement of the basilar membrane. It is known that the basilar membrane is wider at the apex, and the stiffness decreases from the base to the apex. This slide shows you a simplistic diagram of the cochlea when you unravel it from its coiled form. This is probably the best way to show you how movements of the fluids influence the movement of the basilar membrane and how this transduce the auditory signals that we hear in our environment. As you can see here, when the stapes hit the oval window, fluid moves and influences the basilar membrane depending on the characteristic of the sound waves that we detect in our environment. It can lead to the basilar membrane bending in a specific way. It was determined that where the basilar membrane moves can encode for the frequency of the sound that we perceive in our brain. In this case, the movement of the basilar membrane in the base leads to the perception of high-pitched sound, while movement of the basilar membrane closer to the apex leads to the perception of low-pitched sound. The movements of the basilar membrane translate to the movement of the structure in the organ of Corti. If you refer back to slide 30, the organ of Corti lies in the scala media. The organ of Corti is made up of hair cells, classified as inner and outer hair cells. You have the tectorial membrane that connects to the end of the cilia of the hair cells, and movement of the basilar membrane leads to the movement of the tectorial membrane, which bends the cilia, and this is the basis of how action potentials are generated in the cochlea. As you can see on this slide, bending of the cilia starts the process of generating an action potential. You have mechanically gated ion channels present in the cilia, which opens up when the cilia moves. This allows for potassium ions to enter the cell, making it more positive and depolarizing the cell. Depolarization of the cell leads to an increase in intracellular calcium ions, which then triggers the release of the neurotransmitter into the synaptic cleft. Beyond the hair cells, it was determined that one spiral ganglion fibers synapse with one inner hair cell and numerous outer cells. Now, there are functional differences between the inner and outer hair cells. As we discussed in the previous slide, inner hair cells are responsible for transducing an action potential, which is propagated to the brain, while the outer hair cells, on the other hand, acts more as a cochlear amplifier. What this means is that it supports the signals generated in the inner hair cells by influencing the bending of the cilia. Within each outer hair cell, there is a motor protein that enables for hair cells to change its length. This can influence the degree in which the inner hair cell can bend and depolarize in response to a stimuli. The following diagram shows you the main pathway of the auditory system, starting from the receptors. Information is carried by the auditory nerve to the ventral and dorsal cochlear nucleus. This then projects to the superior olive and carried up the inferior colliculus, then the MGN, and lastly up to the auditory cortex. It is important to note that there are other pathways in the auditory in this auditory pathway that are not highlighted in this diagram. The following pathway is the most well-known in the current literature. As we previously discussed, within the basilar membrane, a frequency map can be imagined from the base to the apex. It is important to note that the frequency map is also preserved as information is propagated to the brain. When you have a specific area or neuron that responds highly to a specific frequency, this is defined as the neuron's characteristic frequency. As you can see in the diagram below, the example neuron depicted here is highly responsive to approximately 1,700 Hertz. It is also important to note that neurons in the superior olive, if you refer back to the auditory pathway slide, is considered binaural, which means that they receive input from both sides of the ear. So far in this lecture, we've addressed how we discriminate between different sound frequencies. The question now is, how do we determine the location of the sound? Well, this is done in two ways. Different processes are implemented for localizing sound in the horizontal and the vertical plane. Within the horizontal plane, sound is localized by processing interoral time delay and interoral intensity difference. Interoral time delay is defined as processing the difference in time for sound to reach each ear, and these can be mainly assessed by binaural neurons in the superior olive. Interoral intensity difference is when there is a difference in sound intensity received in one ear over the other. Again, differences are processed in binaural neurons for this process. This slide shows you how interoral time difference and intensity difference work. If you focus your attention to figure A, this is where the sound source come from the right side. If you imagine a sound wave traveling from the right side to the left side, it will usually reach the right ear first, and then it reaches the left ear. In the left ear, you would also notice that there is a sound shadow on the left ear, which contributes to the perception of the location of the sound. Focusing on diagram B, this shows you a similar situation, but in this case, the sound is traveling from the front. You can imagine the sound waves traveling towards the person and how this can create a sound shadow as it travels through the person. The take-home message here is that the sound can be localized in a horizontal plane through the basis of interoral time delay and the intensity difference depicted by this diagram. Now, looking at the process of how interoral time delay is processed in the brain. Taking the example where the sound travels from the left side of your environment, you would imagine that the sound will reach your left ear first. From here, signals will travel through the left cochlear nucleus, and this is relayed to the superior olive. The delayed signal on the right ear will also travel through the right cochlear nucleus and is propagated to the superior olive. As discussed previously, neurons in the superior olive are known to be binaural, which means they process information from both sides of the ear. Action potentials in the superior olive can only occur when it reaches a certain threshold through summation. That is a combination of the right and the left signals. Depending on the summation that occurs in the superior olive, this gives us the perception of where the source of the sound is in a horizontal plane. The sound localization on the vertical plane is more simplistic compared to sound localization on the horizontal plane. From what you can see in the left diagram, the process of the sound localization in a vertical plane is basically done through the structure of your outer ear, called the pinna. The pinna is structured in a way that it can reflect and collect sound into your ear. Depending on the path in which the sound travels into the ear, it can provide the auditory system a perception of the location of the sound in a vertical plane. Axons that leave the MGN project to the auditory cortex via the acoustic radiation. It was determined that the structure of A1, which is the primary auditory area, and the secondary auditory areas are similar to the visual cortex areas. Furthermore, you can see that the frequency mapping is also preserved in the cortex as well, as you can see in this diagram. Studies in the auditory cortex have confirmed the following properties of the auditory cortex. Firstly, you have the tonotopic maps, which were determined with the preservation of the frequency maps from the basilar membrane into the auditory areas. Furthermore, the neuronal organization were shown to be columnar with binaural interaction, which means that both sides of the auditory cortex receives input from both sides of the ear. This leads to the confirmation that unilateral lesion in the auditory cortex can lead to an almost normal function. Lastly, it has been shown that different frequency bands can be processed in parallel as well. The next section of this lecture looks at the vestibular system. The vestibular system plays a crucial role in our day-to-day activities as it is associated with balance, equilibrium, posture, head, body, and eye movement. The vestibular organs are present in the cochlea, which is depicted by the labyrinth structure that you can see in the diagram below. The vestibular labyrinth contains the otolith organs and also the semicircular canals. Similar to the auditory system, action potentials are generated by hair cells present in these organs. The otolith organs play a role in detecting changes in head angle and also linear acceleration. The diagram below shows you how linear acceleration can influence the hair cells, which leads to the depolarization of the cells, sending signals to the brain. The semicircular canal plays a role in detecting head rotation. This has a push and pull effect, whereby both sides of the vestibular labyrinth on your left and on your right will either be activated or inhibited depending on your head rotation. And this is exemplified in the diagram that you see on the right side. So in this case, rotating your to the left activates your left semicircular canal and inhibits the activity on the right semicircular canal. So to conclude this session, hearing and balance have similarities in how they detect stimuli. In this case, if they use identical sensory receptors in the form of receptor cells, which is activated by movement detectors in the form of waves of air pressure, rotational, and linear forces. Where they differ is the source of the stimuli, where the auditory system senses the external environment, while the vestibular system senses movement of itself. Another important thing to note is the parallels of the auditory system with the visual system. What was observed is that the tonotopic maps were preserved from the sensory cells to the cortex in both systems. And lastly, information propagated into the respective primary areas of the auditory and visual system involves the convergence of the inputs, where neurons are considered binaural or binocular, where neurons receive input from both sides of the ears or eyes, respectively. With that said, the lectures has now concluded. So if you guys have any questions at all, feel free to ask them on the chat, and I'd be happy to answer them. So we have a question on the chat. The first one is, I just want to double check something. For the central visual system, does the optic tract leads to the diencephalon? So the answer there is yes. So the diencephalon is division of the forebrain and is situated between the telencephalon and the midbrain. It consists of the thalamus and the hypothalamus as well. And the fact that the retinofugal projection ends up in the lateral geniculate nucleus, which is part of the thalamus, it does lead to the diencephalon. Yeah. Great. All right, if we don't have any other questions, feel free to drop off and I'll conclude the lecture now. If you guys have any follow-up questions at all, feel free to send it to me via email and I will answer them offline. Thank you, everyone.